Yes, excellent. Okay. So let's jump back to, I don't know if I can move my presentation here. Let's jump back to the hatchery, to the Mediterranean hatcheries. The emblematic species are the European sea bass and the gillhead sea bream. And PerformFISH has focused on these two species. Those of you that work in a hatchery, you know that this is a marvelous job because you can watch these eggs starting from very simple structures and then developing gradually but very quickly to proper juveniles that they are subsequently moved to cages, and they start growing, feeding, and then going down to production. However, we also know, after all these years in hatcheries, working in hatcheries, that the hatchery stage is really crucial. And you know that, although you don't know, even you don't know about epigenetics, you know very well that it's often that the whole quality of production, the overall quality of production down to the cages, it's something that it is reflected at the hatchery stage. And you know that you handle very critical developmental stages that can then have very great impact in the overall production. So improving this juvenile quality has always been an internal quest in fish hatcheries, in Mediterranean fish hatcheries, and even starting or being in a position to predict this juvenile quality is going to be a powerful tool. The overall objective of the hatchery stage is the production of high quality juveniles, and that means that we have fast growing and robust adults when they are moving out in the sea cages. So they are juveniles that they have a high feeding efficiency and adults that have high feeding efficiency. And also they are juveniles very robust in health. And also we want to secure the welfare of the animals that we farm. Achieving this overall objective in hatchery, we have tried in this internal quest for improving and predicting the juvenile quality. We all know that there are major influences identified. And this major influence, they start from broodstock and they go down to nutrition. And these are two major influences that they are very well known for so many years. But also we have new things coming up, and this is the microbiome and the abiotic factors. So in PerformFISH, just to give you an overview, we have focused, there are groups in PerformFISH that they've been working with broodstock in sea bream, and they've been working first to improve the genetic background, improve as much as possible, but at the same time, they try to apply programming techniques in order to manipulate the quality of the offspring by intervening at the broodstock level. And you can see there are already publications coming out from PerformFISH, and I have indicated one here for you. Microbiome has also been a new thing for PerformFISH. And if you go back and check the indicated publication here, were published in Microorganisms just at the end of last year, you can see that there is a big fluctuation and there are specific drivers that they shape the microbiome in sea bream and sea bass hatcheries across the Mediterranean. Nutrition has always been the holy grail in hatchery, in Mediterranean hatcheries. We start feeding live feeds, and then there is so much going on about enrichments and how we can really supply for nutritional requirements at these very early stages, that these requirements change very, very quickly, day after day, and how we can get over to successful weaning for these developing animals. In PerformFISH and in collaboration with 4 commercial hatcheries across the Mediterranean, west to east, we have come to realize that there are very fluctuating practices when it comes to early nutrition. Here, for example, in a forthcoming publication, you can see that there are different practices in the use of algae, rotifers, and the duration for how long they offer or rotifers, and then how they get on to feed Artemia and when most of the hatcheries they start weaning. As you can see, the variations are very high. And of course, you can imagine that there are major consequences for later quality in all this variation in nutritional regimes. Temperature has been the major abiotic factor identified as crucial in early development. And this is why Francesc started talking epigenetics and talking temperature right away. This has also a second dimension, a very big dimension, and this is the ecological dimension. Francesc already mentioned that when we're talking epigenetics, this is not confined to farming, but also has a broad perspective because temperature is everywhere. Our animals are ectotherms, and of course, all the studies that they have been using these farmed populations, they are also extrapolated to wild populations. So what we see here has also a very big consequences for wild fish stocks. And in the perspective, in the lenses of the climate change, you can imagine that this takes a completely different dimension, a very profound dimension. So here, we're going to talk out of all these influences that PerformFISH has targeted during these 5 years of implementation. We're talking only temperature and epigenetics today. And this is because I think that we have come to realize that epigenetics is about plasticity. We know already that these early stages are highly plastic. And as you can imagine, epigenetics can, if they are manipulated in a controlled way, we have the feeling, and I think we have come to believe that they can push the boundaries of early plasticity to different levels that they can be predicted, and also to the direction that it is important for farming and robustness and performance at the on-growing stages. Today, I'm going to talk about a specific question, and this question is how the fish quality between the different autogenetic stages it is correlated for sea bream that has been maintained at under different temperature regimes during the autotrophic phase. We are about to see that this early temperature regime has affected the pace of development, how quickly they went over the different developmental stages, the rate of survival, behavioral traits, frequency of deformities, the body shape, the capacity to respond to challenges, and a flavor of molecular mechanism, how the early temperature has affected the energy production. I think that all these here are very important parameters for somebody that works in a hatchery and for producing high quality juveniles that they become fast growing adults. The experiment was a univariate experiment. By saying that, we tried to identify a stage that only temperature would change, and this stage was identified from the epiboly, when the eggs are at a particular stage of development, up to the end of the yolk sac stage. So this is the autotrophic stage, a stage that there are no other manipulation, there is no feed offered to the fish, so there is no other parameter to affect the development other than the temperature. So gillhead sea bream was subjected to 3 different temperatures, 17, 20, and 23 degrees from epiboly up to the end of the yolk absorption stage. And after that, everything went down to 1 temperature, and that was the 20 degrees. So it was a very short developmental window targeted here for temperature variation. And you can see also the recorded temperatures during the experiment. Other than that, it was a standard protocol, a standard rearing protocol, that it was completely dark during the autotrophic phase, and then it went to 24 hours of light. The developing fish, the developing larvae, were fed at the beginning and reached rotifers, and then they went on to Artemia, and the pseudo-green water method was applied to these tanks. Standard zootechnical parameters were recorded, and as you can see here, it was the body weight. We have two different graphs because we have milligrams at the beginning and then grams later on when the fish they start growing quickly, and also the standard length. The major zootechnical parameters did not appear to differentiate with different early temperatures. However, if it took different time for the fish to reach different developmental stages. So it was longer for the fish grew at 17 degrees to reach the first feeding and then the flexion stage. And that also differentiated slightly between the 20 and the 23 degrees when there was a 1 day difference between the two groups to reach the flexion stage. It's very interesting to see how the different groups, they caught up with development. So it was at the same time, at 46 days of development, that they reached the all-fin stage, as we call it. However, survival was not the same. So there was significant difference. There were significant differences between the survival rates when it was measured, assessed at 60 days post-hatch. And for fish grew at 23 degrees, it was significantly lower compared with those grew at 17 and 20 degrees. As you can see here, there was a big differences in survival rate. So we had a major zootechnical parameter affected as temperature increased at the early autotrophic stages. As fish grew, and before they were transferred to sea cages, because that was a full production cycle, as you are going to see later with Debbie Power in the next presentation, they were subjected to a behavioral test. This is called the condition position preference test, and it is a test where the fish, they are challenged to cross over to another side, a side that they don't know. So a construction like the one you see in this photo, where it is a black box really, but with few holes plugged on them, it is immersed in the tank with the fish, and then the test measures how many of the fish they really dare to cross through these holes and explore what lies behind this black barrier within the black box. So in this way, fish, they are the larvae, the juveniles, they are divided into two groups. One is called the bold group, and the other, the one that dare to cross over the other side, and the other is the shy, the fish that they never take this adventure, and they never want to find out what lies behind the barrier. And as you can see here, this test reveals significant differences. Fish that they grew at 17 degrees, there were completely different percentages of bold and shy fish as compared to those grew at 20 and 23 degrees. No particular differences were recorded between the 20 and 23 degrees, but 17 degrees really made a difference in this behavioral trait of the fish, how adventurous they are, and how bold they become in their behavior in the different challenges that they get in the surroundings. Other big experiments, it is about the deformities and the critical swimming speed. So this developing gillhead sea bream at a size of 13 to 15 millimeters of total length, they were assessed for frequency of deformities. And then at a later stage, a few days later, at size of between 18 and 19 millimeters of total length, they were subjected to a challenge called critical swimming. And when they were assessed for deformities, very important and very interesting results were recorded. As you can see here, we had the deformities of caudal fin, head, and then the gill cover were recorded. And significant differences were observed for caudal fin deformities, and those larvae grew at 17, 20, and 23 degrees. As temperature increased, the caudal fin deformities also decreased. That was different from deformities from forehead deformities. And as we can see here, between 17 and 20 degrees, there were no differences. However, 23 degrees really made a difference. There are some details in the experimental design that calls for cautious interpretation of these results. However, this is when it comes to caudal fin, you can see that this has been observed before. So it is like reassessing and reevaluating reassuring results that were first observed and taken from another study. And but when it comes to the head deformities, it is still there's still, let's say, a cautious interpretation of these results. But there is a whole new world to discover if you go and check on this publication that came out, PerformFISH, and contains the results on the frequency of deformities for gillhead sea bream that grew in different temperatures during the autotrophic stages. Temperature definitely affected the body shape. And this is a test, these are measurements based on 8 different points, as you can see here, indicated in the larvae body. And according to these 8 points, then the model can reformulate and can assess the body shape, and also the location of the fins, if we have a different fin location. And it was very interesting to see that fish raised at 17 degrees, again, they were different from those raised at 20 and 23 degrees. They displayed different axis ratio, the body axis, and then also they displayed different positioning of the pectoral fins that are very important for swimming, and also for different mouth positioning. And these characteristics combined, they are characteristics that they favor swimming and preying. Does this really reflect performance? Let's see. Later on, the fish, they were subjected to a challenge, to a major challenge. This major challenge was swimming. Do fish larvae that they grew at 17 degrees really had different swimming capacity? That was the case. So it was very nice to connect, to correlate these differences in body shape with differences in swimming performance. Here, the larvae, the growing larvae, they were subjected at this test, the critical swimming test, that starts with continuous swimming, and the speed of swimming increases incrementally and increases according to total length. And this is why the measurement for the rate of critical swimming starts with total length. So all fish, they started swimming at a speed of 2 total lengths per second. And as they kept swimming, there was an incremental increase of 1 total length per increment of increase. And the fish, they kept swimming through fatigue. So at some point, they were not able to swim anymore, and they were removed from the swimming tank. The results of this challenge test, they revealed that those fish that they grew at 17 degrees, they had the capacity to sustain higher critical swimming speeds. And this was very well connected and correlated with the differences in the body shape, but also we could see differences in the energy production. So the same fish, they were subjected to molecular analysis. This time, we targeted the mitochondria. This is a place within the cell where this energy production factory, it is positioned, it is located. There we have a machine that it is, we have several complexes, several energy production complexes that they work in synchrony in order to ensure that the cell, it is provided, it is supplied with sufficient energy to keep functioning. What we see here are different genes that three different genes that they encode for particles in this complex, in these energy production complexes. And here we see a gene that it is a marker of mitochondria of mitochondria biogenesis. So the increase of this marker indicates that more mitochondria will appear within the cell. It was very nice to see that there were differences before and after swimming. So let me explain these graphs for you. The graphs that they are slashed, they are the gene levels at resting position before the fish they started swimming. So those fishes, those individuals that they appear here in slash bars, they were never subjected to this swimming challenge. And those that they appear in solid bars, they were subjected to the challenge. It was very nice to see that the challenge itself discriminated larvae grew at 17, 20, and 23 degrees. So before swimming, no differences were evident. But when the fish were subjected to the challenge, it was only then that we could see fish at 23 degrees, they had the capacity to start, or they had the need, let's say, to start to increase their gene production, their gene transcription in order to respond to the challenge. That was not the case at 17 or 20 degrees, but at 23 degrees, we could see a clear pattern, as you can see here. And the levels of gene transcription increased significantly between those that they were subjected to the challenge and those that they were not. As we can see here, this is very nice evidence that early temperature has a profound effect on how fish can respond, on their capacity to respond to a particular challenge. This is here, and this is the swimming challenge. And we know that this is a very important function, a very important operation for a fish. And I think that later on, when Debbie Power takes on after me, you can see that also there are other profound effects on other challenges, on how they respond to other challenges as well. Critical swimming, this is a clear measurement, and then very well connected to molecular indicators. If we dive a bit further to these molecular mechanisms of energy production, we can see that the different complexes of energy production, as you can see, they are arranged in the mitochondrial membrane, in the inner mitochondrial membrane, and they consist of a series of complexes. If we go and check, they really look like machines here, and they are machines. If we go and check all the genes that they are responsible for building these machines, and we focus as an example for you in this particular one that they boxed here, and this is called complex 2, we can see that we had different ontogenetic patterns at different temperatures of development. And again, this is only during the autotrophic states. Every box is a gene. Every color is a different level of transcription. Red means we have a high transcription rate, and then as we go to deep blue means that we have a very low transcription rate. And then in each panel we have 3 developmental stages: the first feeding, the flexion, and the middle metamorphosis. As you can see here, comparing the 3 temperatures, there were significant differences between the patterns for each gene at each one of the temperatures. For example, if we go for the first gene here, no need to record anything more about the name of the gene, you can see that we have a higher transcription at mid metamorphosis in those fish grew at 17 degrees compared with the 20 and the 23. And in 23, the transcription rate decreases faster at flexion compared with 20 and 17. And if we go further, you can see that in every single gene, there is a differentiation between the ontogenetic profile at the 3 different rearing temperatures. So we can say that early temperature variation affected the ontogenetic profile of genes encoding for energy production complexes. The same work has been done for all these machines that you see at the top of the panel. We just pick this one that is the one that has the least particles, the lowest number of particles, for you to be more comprehensive. But believe me, if you look at all these kind of panels, we could identify substantial differences in the ontogenetic profile of genes. And I'm going to close with CBAS, just to give a flavor how different parameters of the early development can interplay in order to provide differences in zootechnical and other parameters. 2 different trials were conducted with CBAS. In both trials, the temperature regime was exactly the same. So the eggs, they grew to a single temperature up to epibole, and then they were split in 3 groups. One grew at 14 degrees, 17 degrees, and 20 degrees. And then at the end of the yolk sac phase, they were back to a single temperature, and that was 17 degrees. Exactly the same happened in both trials, but the trials differentiate in the feeding regime. So in the first trial, the first feeding started very late. It was only after 17% of larvae, of developing larvae, they had gone through the shrimp bladder inflation. So that is a very late first feeding. In the second trial, we had a standard rearing protocol with early first feeding before the shrimp blood inflation. The survival rate, as you can see here, it was very low. Of course, this is the combined result of temperature and mainly nutrition. However, under these restricted feeding conditions, we can see that those fish that they grew at lower temperature, they were the ones that they had the lowest survival rate. And it took 17 to 20 degrees in order to have the highest survival under this restricted feeding regime. In the case where we had a proper feeding, a proper early feeding, this difference was not evident. So we can see here that, first of all, it takes more parameters in order to get a result. Temperature on its own does not change everything. So, and we have to be completely aware of this. And the second is that, again, it is a challenge later on that can reveal the result of the early temperature. So at the second trial, where there was proper feeding, proper nutrition regime, 14 degrees did not appear to differentiate in comparison with 17 and 20. It took a feeding challenge in order to reveal what was the effect of the temperature for those fish grew at 14 degrees. So, and also these 2 trials, when the fish they were on and they were subjected to the condition position preference test, the test that differentiates behavior and also differentiates fish between bold and shy, you can see that there are profound differences. It was again the 14 degrees that differentiated, and there were highly bold fish. It was the highest percentage of bold fish ever recording in our trials compared with the shy. So those that they survived, they were really bold compared with the other 2 temperatures. Whereas when we had a proper feeding regime, an early feeding regime, no differences were recording in behavioral traits between the 3 groups. So I'm going to close here with a few messages. I think that there is more evidence. If you go back and check all the papers that we have provided here, you will see that the temperature regime had during this autotrophic stage in gillhead sea bream had profound effects on several zootechnical parameters and behavioral traits and morphology. For example, here I can add the otolith size and shape and symmetry, also differences in the heart morphology and different capacity, as we said, to respond to swimming challenges, and also the gene autogenetic profile of key functions. From this experiment in sea bass, we have come to realize that several factors interplay to define these epigenetic effects, and also take challenges most of the times to reveal these epigenetic effects. But in one way or another, it is important to realize that early developmental stages are highly plastic and they are subject to epigenetic manipulations. This is a great tool. This is a great tool for hatcheries and hatchery managers, because epigenetic manipulations at hatchery stage can become, or is becoming, a whole new world. And this is important because the hatchery can become the precision farming stage in Mediterranean fish farming. And only then we can have predictable juvenile quality at on-growing stages, and really capitalize on our knowledge on epigenetic manipulations. The research on epigenetics is booming, and probably you see that there are so many angles that we try to look at this subject and identify critical time windows and functions that are affecting, but also the underlying molecular mechanisms. So it is important that we know how things work before we can extrapolate to other species, and also before we can gain information from from all these different studies. So I'm going to leave it here. Thank you very much for following this presentation, and I'm open to answer your questions.